Okay, guys, we're going to continue on in chapter 13. I'm going to pick up where we left off, starting section 13.2, entitled Major Factors in the Development of an Infection. And we're going to really explore 5 aspects of infection, and you see those, of course, listed here. First of all, how does a microbe, an infectious microbe, get into you to cause problems? Once inside, it has to have some ability to adhere to your tissues and also survive what you're going to be throwing at them in terms of an immune response, right? And assuming that they can adhere to tissues, survive the effects of phagocytosis and other immune responses, how do they cause tissue damage? We'll talk about that. And finally, we'll also describe some portals of exit, how these microbes leave the body and perhaps go on to infect other people, right? So let's define a few terms, including this first one. What does it mean to be a true pathogen or primary pathogen? Well, this is, of course, the microorganism, be it bacterium, protozoan, virus, fungus. It's the entity that's, of course, causing the infection and the disease in a healthy individual who we will assume has a normal, functional, mature immune response or immune defense system. As opposed to an opportunistic pathogen, which I think you already have a sense of what that gets at. This is another microbe that is taking advantage of a compromised immune system of the host and growing perhaps in a situation where ordinarily they wouldn't have the ability to cause problems. And we talked about this in the previous Zoom lecture, didn't we, when we were talking about how in females, yeast infections can arise in the vagina as a result of antibiotics that were initially prescribed to fight off, say, the true or primary pathogen, and these opportunistic cells, these fungi, these yeast cells, now have unfettered ability to cause problems. Prior to that, they were being kept in check, weren't they? Yeah. Or we can think of somebody, let's say, who has late stage AIDS. Oftentimes, individuals die not as a result so much of the virus itself, but as a result of the effect of the virus compromising the immune system to such a degree that somebody dies from an infection that you and I would have no trouble fighting off, but because their immune system is so depressed that it overwhelms the immune system and the person passes away as a result of the pneumonia or whatever the opportunistic pathogen, whatever it causes. So I think this table sort of is something that you and I could come up very easily if we brainstormed and we thought about, well, what factors could suppress or weaken an immune system of an individual, or what might make somebody more susceptible to getting an infection and a disease. Well, obviously, these 7 bullets kind of address that. The fact that as you get older, or if you're extremely young, if you're a premature baby, right, your immune system may not be fully up to par yet. Maybe the lungs aren't fully functional yet. And as a result, pneumonia may be more common. Genetic defects, AIDS, this depression of the immune system as a result of the viral infection, or somebody maybe is born with a genetic mutation that predisposes them to a non-functional or significantly compromised immune system, right? Having surgery, organ transplantation is fraught with potential rejection issues and complex follow-up infections, opportunistic infections. Anytime you introduce an incision in the body, you introduce the capacity or possibility of pathogens getting in, right? If you're fighting off another illness, cancer, heart disease, diabetes, obesity, these all significantly increase the risk of problems down the road. Being under a chemotherapeutic drug or immunosuppressive drug as a result of having cancer can significantly increase the risk of infection. Even physical and mental stress has been shown to increase the risk of infection by weakening the immune system. The importance of getting adequate sleep at night, running ragged does often result in a person getting sick, more likely, because of the stress we place on our organs or our organ systems. Virulence. What does that mean when we talk about the virulence of a microorganism? Because some pathogens are more virulent than others. So the severity of the disease depends upon how virulent the pathogen is. And different pathogens have different virulence factors at their disposal to cause infection. We'll be talking quite a bit about some different virulent factors in just actually a few minutes. Well, let's begin our survey that we introduced a few minutes ago as we look at the evolution of an infection, starting with the need of the microbe to get into the body. And I think, again, a lot of this is pretty self-explanatory and directly relates to where we tend to find pathogens, both, I should say microbes, both pathogenic and non-pathogenic types. We talked about that again early in this chapter. Portals of entry. Obviously, skin is an important portal of entry. We'll talk more about that in just a minute. But it's not the only way that things can get in. Oftentimes, when we talk about exogenous microbes, we're talking about the fact that these microorganisms originated from outside the body. If you inhale droplet nuclei that are containing COVID-19 virus, and you inhale that through your respiratory tract, that's an example of an exogenous agent. So we try to wear masks, we try to social distance, we attempt to minimize the likelihood of acquiring that respiratory virus because we know it's out there, right? Endogenous, as the name implies, is coming from something already there or in our body. So that secondary infection that we described earlier with regard to yeast infection, the opportunistic candida, causing problems, that already existed in the vagina, didn't? But as we said earlier, that was kept in check by the residents. But if we kill the residents off, then these opportunistic endogenous fungi can cause problems, i.e. yeast infection. Okay, so we're going to briefly talk about 5 different portals of entry. Starting with the skin, we know again that the epidermis, being extremely thin, is an effective barrier. But if we break through that very, very thin layer of the skin, and we've all had cuts and bruises and bumps and gashes, we introduce the possibility of entry. When we eat or drink or ingest materials, sometimes they can carry pathogens that can cause gastrointestinal upset, right? We've all experienced probably a time when we ate at a restaurant, we came home later that day or even that evening and got sick. And it wasn't because we acquired a virus, but was because probably we ate some bacteria that was laden in the food that we were served. I think we've all had that happen to us on occasion. Cholera, a real problem in many parts of the world that are suffering famines or that have undergone a natural disaster, and the water gets contaminated with fecal material, and you drink that water, you can come up with this very, very terrible dysentery. There are other ways we can get this too, right? We can drink water that has Giardia in it. Remember Giardia, that protozoan we talked about earlier in lab, I think our second lab. We also referred to it back in the chapter that surveyed different kinds of eukaryotic pathogens. We just mentioned a few moments ago, COVID-19. Many of the flu viruses enter through the respiratory tract. They get in through our nose, through our mouth, and eventually get into our bloodstream and cause problems. Urogenital tract. This is sort of a combination of the urinary and the reproductive tracts. And the table actually shown here addresses a little bit of common sexually transmitted diseases here in the United States. And you'll note that some of the sexually transmitted diseases that we might be more familiar with, like syphilis and gonorrhea, aren't the most common in terms of numbers of cases in a year in the US, but rather it's viral or protozoan related STDs. So human papillomavirus or genital warts, 14 million new cases a year in the United States. A little over 2 million trichomoniasis infections. This is an infectious protozoan we talked about in one of the earlier chapters. So this continues to be a major issue. And again, these are transmitted typically through the urogenital tract as a result of sexual intercourse. Transplacental is, of course, getting at bacteria or viruses or other infectious agents that can cross the placenta or enter the baby at the moment of birth or during the birthing process. And important to also make note of the fact that even though we address these 5 separate portals of entry, it's possible that, as it mentions here, some microbes, some infectious agents may have more than one portal. And as a result, how that is presented in terms of symptoms of the patient can vary significantly. And so a physician needs to be able to assess those symptoms and do some brainstorming here. Maybe it's the same bug, but the symptoms are indicating a multiple portal of entry situation. So I want to just take a few moments and talk about the transplacental portal. There's just a short blurb there in your book, but I think it's quite interesting. Talking about pathogens that infect during pregnancy and birth. And it's interesting to note that there are a few microbes, such as the syphilis spirochete, that can cross the placenta, enter the umbilical vein, and spread into the fetal circulation and get into the fetal tissues. The placenta is generally a fairly important, effective barrier to most microbes, but things like, as we mentioned, that spirochete, that prokaryote, can get in beyond and past the placenta. So we talk here about STORCH, this acronym, of course, which you can see here in front of you, standing for a variety of disorders, diseases, infections. A lot of these are viral, aren't they? Some of these entities develop perinatally when the infant is contaminated by the birth canal. So as the birthing process occurs, the individual is inoculated with what mom has, if you will, and the infant can acquire those infections. And so nurses and pediatricians are always on the alert for these STORCH infections. Infectious dose, or ID, is an interesting concept that relates to the minimum number of microbes needed to induce an infection. And we should probably say from the outset that while we give infectious dose values, keep in mind that this could, to a large extent, be impacted by, as we said earlier, the health of the individual. It's his or her age. Is he or she fighting off a pre-existing disease? So there are lots of variables here that one needs to be mindful of and take into account as we assess these various infectious doses. But it's an interesting and an important concept to be aware of. And of course, you see in this table a list of some different diseases, infections, right? Their infectious dose estimate and their portal of entry. How do they get into the body? Then you'll notice that some of these diseases, like measles or shigellosis, have very low ID values. All you need is one viral particle to get measles. It's extremely contagious. Tuberculosis also. Norovirus, which causes, lots of gastrointestinal upset. Smallpox. We're talking just a few cells or a few viruses. It's all it takes, assuming it gets into the appropriate proper portal of entry, can cause infection and disease. But even some of the more weakly pathogenic species, and we can drop down sort of to the bottom of the list as we encounter anthrax, that's a bacterial infection, or cholera, as we've talked about, it has a fairly high infectious dose. But when we ingest, for example, cholera in large numbers, and it's very easy to do that, large doses of relatively weakly pathogenic low ID, or I should say high ID values, can be extremely dangerous. Okay, so let's assume the pathogen has gotten in. How might it attach itself? How might it gain a foothold in you and I? Well, of course, that depends upon the microorganism, but it has to have some way of attaching itself to our tissues, to our cells. We've already talked about some of these, so this is pretty much review. The fimbriae we talked about in one of the earlier chapters. These can help the cell, we said, attach to some substrate, and here we see some bacteria with some fimbriae that are attaching to the surface of a host cell. The fact that even some protozoa, and the one that I'm thinking primarily of is trypanosoma, which causes African sleeping sickness. You might remember trypanosomiasis from lab. Well, it uses its flagellum to penetrate the host cell. Isn't that interesting? Capsules. Well, we know that capsules do impart increased pathogenicity to a bacterium, for example, because the immune cells, the phagocytic white cells, have a heck of a time phagocytizing encapsulated bacteria. But we should also recall, I think you do if you think back to lab, we talked and also lectured, that capsules can help a cell stick to the host cell. And we see that, of course, illustrated here in the middle of the diagram. Viruses, how do they adhere to host cell membranes? Well, you remember from the earlier chapter that they have these glycoprotein spikes that have to fit into receptor sites, right, within the cell membrane of the host cell. They have to have those proper shaped receptor proteins. And once that docking process takes place, the enveloped virus, like we see here, is allowed into the cell. Yeah. When we think about suckers and hooks and barbs, we're talking more about helminth worms, like tapeworms and flukes and roundworms. And this is, of course, a magnified electron scanning micrograph of a tapeworm. And we can see some of the suckers here on the side, and look at the hooks that again help the tapeworm stick to or attach to the lining of your small intestine, perhaps. Yeah, so cells or organisms have all sorts of tricks up their sleeve to help them adhere to the host cell. And this is simply an interesting table that you can kind of take a look at. You'll see some common microorganisms here that you've heard of before, what diseases they cause, and exactly how do they adhere to the cells, to the tissues. And I think this would be certainly worth making note of and looking through. I don't mean you have to memorize the entire table, but look at a few of these. Be familiar with 4 or 5 different microbes, what disease they cause, and how they stick. Okay, let's assume they've gotten in, they've adhered to our cells, our tissues. We're going to try to knock them out. And this, of course, gets actually to the next chapter, which is talking about host defenses and innate immunities that we're going to make use of to try to hopefully knock this thing out. And maybe we will be successful in doing that, maybe we won't. So how do microbes, pathogens, survive these host defenses? Because certainly the white blood cells, the phagocytic cells, are going to try to get rid of those. Well, some pathogens have anti-phagocytic factors or chemicals that they will produce. And we talk here about staph and strep producing these leukocidin chemicals. Leuko refers to white cell. Cidin means to kill. So these leukocidins kill white cells. Yeah. We've also talked earlier about how some bacteria produce capsules, as I mentioned just a moment ago, or perhaps a slime layer, both of which make phagocytosis a heck of a lot harder by neutrophils and macrophages and other phagocytic white cells. Strep and Salmonella are examples of cells that produce these structures. And what is even more interesting to me, I think, is the fact that even if you are phagocytized, if you're Mycobacterium or Legionella, you can actually survive having been phagocytized. Yeah, they don't die as a result of being phagocytized. They stay alive in the white cell. They stay viable in the white cell. So a fascinating biology of this particular set of genera. Some have the ability to actually penetrate host cells, getting in now to our host cells, into our deeper tissues. Salmonella, you've all heard of salmonella or salmonella poisoning, right? This is on the news every few months when you hear about lettuce or spinach, or often it's a green vegetable of some sort, and salmonella has occurred, and many people have gotten sick and maybe even died, and they try to pull this off the shelves, right? So what salmonella does is it actually adheres to the lining of our small intestine, so it's typically acquired through the foods we eat. And what it then will do is it will produce these proteins that will help to actually digest some part of the cell membrane here. And then what the cell does is it actually wraps around the invading pathogen. Okay, so when we talk about villi, these really, you remember, are on the outer surface of the small intestinal lining. The cell is actually pulled into the cell, or the microbe rather is pulled into the host cell, actually becomes part of a vacuole that the cell produces. It's like a little carrier structure where it can undergo binary fission, as we see here. It starts to divide and multiply inside this vacuole, inside the intestinal cell here of you, and eventually it can make its way past that simple columnar cell that lines the mucosa, perhaps, and then can get down into deeper tissues and perhaps be carried away by the bloodstream and set up a more systemic infection within the organism, within you. So it's got a lot of interesting tricks up its sleeve, doesn't it? Salmonella does. Well, how do they cause problems? We know they can sometimes evade the immune system. They have ways of infiltrating cells, getting into deeper tissues. Well, how do we actually get sick? What causes tissue damage? Well, remember we talked about virulence a few moments ago, and we said we were going to talk about some virulence factors coming up. Well, here's that part of the lecture where we talk about how severe the infection is based upon the degree to which these virulence factors are being produced by invading microbes. And different microbes have different sorts of factors. One type of virulence factor that some pathogens make use of are enzymes that they secrete externally, so-called exoenzymes. And so here we've got some host cells. We'll have bacteria colored here in green. And as a result of these enzymes that have been secreted by the pathogen, by the bacteria, the action is to dissolve this initial barrier that existed, right, between the outside of the tissue and the inside of the tissue. And so here we've got this dissolvement, if you will, or the dissolving of cell membrane, and the bacteria now can spill in between the cells and get down into deeper tissues. These hyaluronidase enzymes are notorious for that. The capacity to make toxins at the site of multiplication, or toxogenicity, can sometimes occur as a result of these exotoxins being produced. So here again, we've got bacteria shown in sort of a dark green. And what's taking place? Well, the release of these exotoxins is actually causing the cells to slough off, as opposed to dissolving the gaps here between, or producing the gaps between these cells. So the cells are actually sloughed off as a result of this toxin action again. So different exotoxins can have different impacts, but the overall end result is the ability of the bacterium or its pathogenicity factors or virulence factors, allowing that to get into the tissue. We're going to talk about two specific types of toxins. The first, the endotoxin. Now, we've already talked about the second one, which is exotoxins, just a few moments ago, but let's set the stage for what the difference is between endo and exo. We've already mentioned in the preceding two slides that exotoxins are actively secreted by the bacterial cell. We're going to assume bacterium here because viruses don't produce endotoxins or exotoxins, neither do fungi or protozoa. So this is strictly bacterial. And we've already seen one impact of exotoxins. We'll talk about a few more in just a moment. And let's compare and contrast this actively secreted protein with endotoxins that some bacteria make use of as a virulence factor. And so as it mentions here, these toxins are not secreted, but rather are released after the bacterium is damaged or lysed by the immune system. So yeah, some bacteria can be phagocytized and damaged, but that can induce the release of these endotoxins, which cause us harm. okay? And what's interesting to note is if you think back to our discussion of prokaryotic cell biology in one of the earlier chapters, we mentioned that this outer lipopolysaccharide layer, which is common, of course, in gram-negative cell walls, this exterior part of the membrane, that's actually the endotoxin part of the cell that's causing the problems. It's part of that lipopolysaccharide. Kind of interesting. So we'll talk more about endotoxins in just a moment, but I want to go down to the bottom of this slide, back to exo for just a moment, and make comment about the last two bullets, the hemolysins and the AB toxins. So these hemolysins, as the name sort of implies, these are chemicals that lyse red cells. They cause the red cells to pop. Not good for you or me, right? And the AB toxins, I'm going to let you simply watch a video coming up in a few slides here that kind of talks a little bit about the two parts of this chemical called an AB toxin. One's an active part, the A part as you see here, and the B part is that part of the molecule that binds to the cell membrane before the A part, the toxin part, actually enters in. But these, again, are actively secreted by the pathogen. So one thing that we kind of think about with regard to general systemic symptoms of somebody who's suffering from exotoxin versus endotoxin effect is the fact that exotoxins tend to have more targeted impacts on organs or organ systems. And this is denoted here on the left side. Where we have effect on specific organs like the heart or the hemolysins targeting the red cells, or causing cells to slough off the lining of the small intestine, right? These are localized effects for the most part, as opposed to endotoxin symptoms, which are more systemic, more general. So aches and pains and fever, and overall lethargy would be more indicative of endotoxin effect. So these again are helpful to the clinician as he or she attempts to identify what might be going on in the patient. This was that video that I was referring to just a few moments ago. I'm not going to play it, but I do want you to watch it. So you can go into the PowerPoint and watch that.
AB toxins are
This is a good table, I think, that sort of reiterates what the previous figure was showing in terms of general effects of these two bacterial toxins. Take a few moments and look down the column here as you compare and contrast exo to endo. And I'm going to just allow you to read over that slide. Okay, let's talk next about some of the different stages of an infection as it might occur in you or me. The first stage of which sort of makes sense, the incubation period. This, of course, is that initial stage of an infection, which is from the time you're first exposed to the pathogen to the time you begin to see some of the very, very first initial symptoms. And it's during this time that the infectious agent has penetrated the barriers that we have, our skin, for example, or has survived the action of the phagocytic cells. But we don't really know that we've been infected yet. And it's interesting, as it notes here at the very end of this short little blurb about an incubation period, that in some instances, this could range from just a couple hours from the time you're exposed to the time you're starting to begin to feel some initial symptoms, or Sometimes incubation periods can last for many, many years. You may be exposed to a virus or a bacterium, in this case for leprosy, and you might not know you have it in your body. It could be years later that you start to exhibit symptoms. So it's kind of interesting from that standpoint. It just depends upon the pathogen. But following that initial incubation period, we have what's referred to as the prodromal stage. And it's here that we start to think about, something's just not quite right. I don't feel quite right. Something just feels out of whack, right? Hard to put your thumb on it, your finger on it. You're not sure if it's a general stomach upset or if it's going to result in a fever coming in a few hours. It's just something's going on. And of course, if we were to think about what's going on in terms of number of pathogens in our body, we would know. Think back to that TED talk that was covered in one of the earlier chapters that Bonnie Bassler did on biofilms. It's during this time that the bacterium, we'll just pretend it's a bacterium, is beginning to mount its attack, right? The numbers of bacteria are going up during the prodromal stage, and you're starting to feel some of these initial symptoms. Well, it's the period of invasion where we have maximum cell division taking place. The pathogen has initially, hopefully temporarily, overcome the effects of the immune response, and you are feeling sick. You have fever, you have dysentery, you are throwing up, you are just down and out, can't do much of anything, right? With a peak of infection arising at some point during this stage. And of course, how long does it take to go from incubation to the prodromal to the infectious invasion timeframe? Really depends on lots of factors, right? On the health of the individual. What is the microbe? What is it throwing at us in terms of its pathogenicity? Does it have endotoxins it's producing or exotoxins? So this can, of course, vary a lot. But at some point in time, we hope our immune system is able to take charge and knock that pathogen out, and we begin to feel better. The convalescent period, right? So these are just general stages of a typical clinical infection, but certainly worth looking at. Let's talk about some other terms that are described in the patterns of infection section of the text. The first one is localized, which simply means at one particular region, it's confined to a particular part of the body. Now, that may be where the microbe entered. It may not be, but often is. So if you get a cut on your arm or your finger, let's say, and sooner or later you start to see some swelling and you have some tenderness right at that infectious site where that bacterium was introduced, that's a localized infection, right? As opposed to systemic infections, meaning that this is spreading throughout the entire body via the bloodstream or perhaps even the cerebrospinal fluid, right? Often it's bloodstream. And so this means that pathogen can get all over the place. Systemic. Focal. Focal infections occur when a local infection spreads. So we start off at a localized region where that infection occurred or began, and then the pathogen breaks off, if you will, or disseminates to other areas of the body. What's really kind of interesting is the fact that you can have an infection site at location A, where the bacteria is, where it's growing rapidly. You have lots of cells that are producing exotoxins that are carried through the bloodstream, but whose target tissue is far from the initial localized region. And your book makes an example of how a streptococcal infection in the pharynx can give rise to scarlet fever, which impacts the heart. So don't always assume that the infection is right at that same location. It can very often be elsewhere in the body. Mixed simply means you have more than one species of microorganism that are causing problems at the same time. They're simultaneous in terms of their impact. Two different species of bacteria. It could even be a bacterium and a virus, theoretically. We know that biofilms harbor a variety of different species of bacteria. Sometimes you can have prokaryotes and eukaryotes living together in a biofilm too. So they can be two different cell types. One prokaryotic bacteria, one eukaryotic, some protozoan or fungus. And we talked about how drugs can work synergistically, if you recall from the earlier preceding chapter. But here we're talking about how the pathogens can work together synergistically, meaning that, you know, combining their forces can be more beneficial to them as invaders, meaning you suffer the ill effects of both combining their forces, causing issues with you and me. And that can sometimes happen. Primary and secondary, I think that's pretty self-explanatory. Primary, again, initial infection at that site. A secondary infection at some other site somewhere else, perhaps caused again by a totally different microorganism. Here we're talking about a viral infection, chickenpox setting up in one location, but allowing Staph aureus to become the problem microbe at that particular site, perhaps, or elsewhere in the body. And typically, we can also think of this, I guess, as sort of a, well, secondary infections can set up at a site different from where the primary one does. And as it mentions here, this can be often due to a weakened immune system of the individual. Acute and chronic. Acute, just getting at the idea that the infection comes on rapidly, and typically it hits you like a train. The upside, not that this is always the case with acute infections, but oftentimes it hits hard, but the symptoms are rather short-lived, as opposed to a chronic infection, which can last a long, long, long time, and the symptoms can be more persistent. Some warning signals that we associate with different kinds of diseases. I think we've all suffered from these sorts of symptoms, right? Chills, fever, nausea, stomach upset, stomach cramps, headache, all indicating what could very well be, and likely is, some sort of microbial infection. Right? Some signs and symptoms. This really truly gets back to A&P. So I'm not going to spend a ton of time on this, but we've talked about how one of the earliest symptoms of an infection is inflammation, right? Swelling due to accumulation of fluid, edema, or walled off zones that contain microbes, these granulomas and abscesses. The body attempts to wall them off, preventing them from moving beyond that initial site. Lymphoadenitis. We've all noticed sometimes we have swollen lymph nodes under our neck, perhaps, or in the axillary region, indicating the immune system is fighting off some pathogen. So this is all stuff you should have learned about in A&P 1. And likewise, some of these other terms that relate directly to white cells or leukocytes the notion that some infections can result in an increase in the number of white blood cells present. Sometimes they can actually cause a decrease in white cell counts. This is all the more important if an individual is suffering from an infection or a disease. They'll want to do a blood draw and take a look at a differential white cell count, where they look at the percentage of the different kinds of white cells. And sometimes you can see elevations of eosinophils or a depression in the lymphocyte count. All these are important clues to the clinician as to what might be going on. What's the pathogen perhaps that's causing the problem? The presence of bacteria in high numbers in the bloodstream. Now, that may very well indicate a particular barrier has been broken and the bacterium is getting in and spilling into the bloodstream, right? But sometimes we don't necessarily find great numbers of bacteria or viruses in the blood, maybe small in number, so that bacteremia and viremia terms kind of describe that. It would certainly indicate that there has been an invasion of some sort taking place, right? And finally, the last part, if you will, of our evolution of the infection is, how does the microorganism leave? Well, if you look at the portals of exit, what do they look like? They look similar to the portals of entry, don't they? Yeah. Most pathogens leave by a specific portal. And this can, of course, greatly impact and influence the sorts of symptoms that the patient is exhibiting. A lot of pathogens, of course, viruses in particular, take the respiratory route. Not all, but many of them do. The flu virus, for example. COVID takes a respiratory route in and out. Other bacteria might be shed through skin. You're losing your epidermis, layer of your epidermis, the stratum corneum, constantly. Well, you're also not only shedding skin, you're also shedding microbes. Yeah, you are. Fecal exit, we obviously know that the feces are 30% by volume made of bacteria. And if we don't properly treat wastewater, if it gets into our drinking water, we can get sick. We see that happening all throughout the world. We just don't appreciate it here in the United States because we have effective wastewater treatment plants, or we have a septic tank in the backyard buried somewhere, right? But in many parts of the world are drinking water that's contaminated with coliform bacteria. And that causes things like cholera and dysentery and other problems. Some microbes leave the urogenital route, sexually transmitted diseases. How are they transmitted? They're transmitted as a result of picking it up from somebody you're having sex with, right? Removal of blood. IV drug users can pass on infections, right? Via the use of contaminated needles. HIV, hepatitis, all transmitted by sharing needles or through small abrasions in the mucous membrane during intercourse. Persistence of microbes in pathologic conditions. It's important to note that generally, most of the time when we go through those 4 stages of the infection and we get to the convalescent stage and we feel better and a few days go by and we're kind of back to normal, right? For the most part. That's not always the case for all microbes. You may still be harboring the effect of that microorganism, right? It's possible that you may get infected and you may get sick and you may get better, but down the road, because you've harbored that microorganism, you fought that initial battle and won, but the microbe can go into a latent stage or phase. And this is common in certain kinds of viruses that get into our nerve cells, for example, and lie dormant for many, many, many years. And somebody who gets shingles, for example, may have had a mild case of chickenpox as a kid, been inoculated with that virus and gotten sick, or maybe had a very, very minor mild form of that particular disease. But then 20, 30, 40 years later, gets a really, really bad case of shingles. I mean, really bad. You can get super sick with it because you harbored that in that latent stage. And as a result of some stress or some physiologic episode that triggered the virulence factors in that virus, it then reoccurs. This is again common in malaria, where a person can get it, come down with the symptoms of malaria, which are the periodic fevers and so forth. And these can last a long time. You can get over it, think you're fine, only to have symptoms again months later. Herpes simplex, good example of another virus that can lie dormant, can lie latent. And during that latency period, you might feel fine, but you might likely still have the potential of shedding that infectious agent to somebody. And they could get sick. So we would call you a chronic carrier. You're possessing or harboring the microbe, and you're shedding it either, again, through respiratory droplets, perhaps, or through skin, or any number of different portals. That's what a chronic carrier is. And your book introduces another term that I've never ever heard of before, other than in this particular text. It's pronounced sequelae. Sequelae. Some diseases leave sequelae, long-term or permanent damage to tissues or organs. And as I thought about this, I added COVID-19 to the list because I think it's a classic example that we can all understand. Because if you've been studying COVID-19, and who hasn't heard about it in the last 15 months, we talk about the long haulers, people who have gotten over maybe the worst part of COVID-19, but are still sick. They've had heart damage. They've had neurological damage that could follow them for the rest of their lives. We don't really know, do we? But it would not surprise me at all to learn that somebody who got COVID-19 at age 50 might continue to have these issues for the rest of their life and could potentially pre-expose them to other illnesses down the road. We simply do not have enough data. Not enough time has expired or transpired, I should say, for us to follow that. But I can guarantee you, we will be following thousands and hundreds of thousands of patients, and we'll have a lot more to say about this as time goes on. But we look at someone who's had Lyme disease, who's been bitten by the tick that harbors the bacterium that causes that nasty disease. Arthritis is often a common problem for the rest of their lives, or somebody who's had meningitis and can no longer hear because of the effect of that disease. So it's just very, very interesting. It's also very sad to think about the fact that while you think you might be over a disease, there are certain kinds of pathogens that can leave scars, both physical and mental, and cause problems that may or may not be overcome by the patient in the near term or even long term. Okay, I'm going to stop here, and we'll wrap up the chapter talking about epidemiology.